Module 7, Objective 11, explain how memories are created, stored, and recalled, and distinguish among the levels of consciousness and unconsciousness. Higher order functions share three characteristics. One, they require the cerebral cortex. We know that the function of the cerebral cortex houses a lot of the higher order functions that we associated with humanness. Two, they involve both conscious and unconscious information processing. And three, they're subject to adjustment over time. So they're not innate, fixed behaviors, but they do change over time. Memories are stored information gathered through experiences. Fact memories are specific bits of information such as the color of a stop sign or the smell of a perfume. Skill memories, on the other hand, are learned motor behaviors. You can probably remember how to light a match or open a screw top jar, for example, and we do that through repetition. Skill memories through repetition become incorporated at the unconscious level. Skill memories related to innate behaviors such as eating are stored in the appropriate portions of the brainstem, while complex skill memories such as how to ski or play the violin involve an integration of motor patterns in the basal nuclei, the cerebral cortex, and the cerebellum. Short-term memories don't last long, but while they persist, the information can be recalled immediately. Short-term memories contain small bits of information, such as a person's name or a telephone number, and through repetition, through repeating a phone number or other bits of information, it reinforces the original short-term memory and allows it to be converted to a long-term memory. Long-term memories last much longer. In some cases, they may last your entire life. The conversion of a short-term memory to long-term memory is called memory consolidation. There are two types of long-term memories. Secondary memories. These are long-term memories that fade with time and may require considerable effort to recall. Or tertiary memories. These are long-term memories that are with you a lifetime, such as your name or the contours of your own body. These all exhibit in the tertiary memory. Here we see a diagram that sort of outlines the different types of memories. We start with sensory input, and as the brain interprets that sensory input, we develop short-term memories. We can promote retention of those short-term memories through repetition. And of course, the more repetition that occurs allows for consolidation of short-term memories into long-term memories. And of course, all this can be lost at any point within this process through lack of repetition. Brain regions involved in memory consolidation include three main. You have the amygdaloid body, or the amygdala, as well as the hippocampus, the nucleus basalis, as well as the cerebral cortex. The amygdaloid body and hippocampus are components of the limbic system. You should think emotions. And these are essential for memory consolidation. If you ever experience damage to the hippocampus, it causes the inability of the conversion of short-term memories into long-term memories. So while existing long-term memories remain intact, they can no longer convert short-term to long-term. I think there's been several movies that were created based on this concept, 50 First Dates, where the individual could never convert short-term to long-term memory, so they relived the experience over and over again. In the cerebral cortex, we find the storage of long-term memories. Both conscious motor and sensory memories are referred to the association areas. The temporal and occipital lobes are special portions that are crucial to the memorization of faces, voices, and words. And specific neurons may be activated through a combination of sensory stimuli associated with particular individuals. We call these grandmother cells. In addition, we have the visual and auditory association areas, the speech centers, the frontal lobes, and related information stored in other locations. And if these particular storage areas become damaged, the memories that are stored in those areas will also be damaged or incomplete. The cellular mechanisms of memory formation and storage involve both anatomical and physiological changes, both at the level of the neuron as well as the synapse. And there are three ways in which these changes occur. The increased neurotransmitter released. Frequently active synapses increases the amount of neurotransmitter it stores. So the more that a synapse is utilized, the more a neurotransmitter is stored within that synapse. And of course, the more become released upon stimulation. And the more that are released, that has the greater effect on the postsynaptic neuron. Facilitation at synapses. Neural circuits repeatedly activated lead to facilitation, and synaptic terminals will begin continuously releasing neurotransmitter. Neurotransmitters bind to receptors on the postsynaptic membrane, producing graded depolarizations. And this is going to bring that membrane closer to the level of threshold and leads to facilitation. So facilitation results affect all the neurons within that circuit. The formation of additional synaptic connections. When neurons are repetitively communicating, the axon tips start to branch. So if you consider a generalized neuron, the axon tips start to branch. And as they branch, they're going to form additional synapses on that postsynaptic cell. And the presynaptic neuron then has a greater effect on the transmembrane potential of that postsynaptic neuron. The cellular mechanism of memory formation and storage. We have the basis of memory storage, processes that create anatomical changes that additionally facilitate communication along specific neural circuits. When you have a single circuit that corresponds to a single memory, this is referred to as a memory engram. And this is going to form as a result of experiences through repetition. The efficient conversion of a short-term memory takes about an hour, but repetition is needed. Factors of conversion include the nature, the intensity, as well as the frequency of the original stimulus. When you have strong experiences that are repetitive, or exceedingly pleasant or unpleasant events, you have the likely conversion of short-term to a long-term memory. I always like to use the example that when a traumatic event happens, and the one that is often cited in textbooks is the death of President John F. Kennedy. People of that generation, they know exactly what they were doing, where they were doing it when they heard the news of the death. It was such a traumatic event for our nation that unpleasant event caused the conversion of a short-term memory into long-term memories. Some drugs stimulate the central nervous system to help enhance memory consolidation through facilitation. These include caffeine and nicotine. A particular receptor type found within these regions of the brain that are called NMDA for N-methyl D-aspartate is linked to consolidation. These are chemically gated calcium channels that are activated through the neurotransmitter glutamate. When glutamate binds, the gates open, calcium enters the cell, and you get the facilitation of memories. Blocking these receptors in the hippocampus prevents long-term memory formation. States of consciousness. There are many gradations of states. or the degree of wakefulness indicates levels of ongoing activity within the central nervous system. When abnormal or depressed, states of wakefulness is affected. Deep sleep, this is also called slow wave sleep, occur when the entire body relaxes. The activity within the cerebral cortex is minimalized. Heart rate, blood pressure, respiratory rate, as well as energy utilization decline up to 30%. Rapid eye movement, or REM sleep, is when active dreaming occurs, and this experiences changes in blood pressure as well as your respiratory rate. It's less receptive. Individuals who are in REM sleep are less receptive to outside stimuli than compared to deep sleep, and the muscle tone decreases markedly. Intense inhibition of somatic motor neurons leads to rapid movement within the eyes as the dream events unfold, hence the name rapid eye movement sleep. Our nighttime sleep pattern, we alternate between various levels. We begin in a deep sleep, and then REM periods average only about 5 minutes in length, and they can increase to 20 minutes over the course of an 8 hours, just depending on how much sleep an individual gets. So our patterns go in and out from just deep sleep into various states of REM where we dream, and it is known that individuals normally dream many times per night, but again, these dreams are very short in length, from 5 to 20 minutes in length, and often people have variable amounts of recall of those dreams. So some folks recall dreams very vividly, while others lack that ability to recall dreams. Sleep has an important impact on the central nervous system and produces minor changes physiologically through various organ systems. Protein synthesis in neurons increases during sleep, and extended periods of time without sleep leads to disturbances in mental function. I know some of the tactics used by some factions in which they don't allow individuals to get adequate sleep, and it definitely affects their mental functions. We've often, probably all, experienced times in which we didn't get enough sleep and we just didn't feel like we were firing on all cylinders. 25% of the US population experiences sleep disorders. Here we see an EEG associated with various sleep states, from the awake to the REM sleep, where one is dreaming, to the deep sleep, and we can see changes in the electroencephalogram associated with those patterns. This is a typical pattern of sleep stages in a healthy young adult during a single night's sleep. You can see they fluctuate from REM sleep to a transitional period that leads to a deep sleep, and then of course they're going to zigzag all through that activity. One thing to note is that the REM sleep periods, they do get longer as the night progresses. Arousal and the reticular activating system, the RAS system, causes awakening from sleep. The functions of the reticular formation, these are extensive interconnections with sensory motor, integrative nuclei, and pathways along the brainstem, and they're determined by complex interactions between the reticular formation, and the cerebral cortex. The reticular activating system is an important brainstem component. There's a diffuse networks and reticular formation extending from the medulla oblongata into the mesencephalon, and the output of the RAS projects into thalamic nuclei that influence large areas of cerebral cortex. When the RAS system is inactive, so is the cerebral cortex. So stimulation of the RAS produces widespread activation of the cerebral cortex. At the end of our sleep period, any stimulus that activates the reticular activating system causes arousal, and this can be very rapid. The effects of a single stimulation of RAS last less than a minute. Activity in the cerebral cortex, the basal nuclei, and sensory and motor pathways continue to stimulate RAS. After many hours, the reticular formation becomes less responsive to stimulation, and the individual becomes less alert and more lethargic. Neural fatigue is also something that reduces the RAS activity. The regulation of our awake-asleep cycles involves an interplay between the brainstem nuclei that uses different types of neurotransmitters. Groups of nuclei stimulates the RAS with norepinephrine and maintains the awake alert state. Other groups promote deep sleep by depressing the RAS actively with the neurotransmitter serotonin. So there's sort of this dueling nuclei located within the brainstem that really is dependent upon the types of neurotransmitters that are being actively released. This particular diagram looks at specific sensory input and the general cranial or spinal nerve input, and the flow from the brainstem into the various regions of the cerebrum.